So we're going to talk about lipidomic data analysis, and I would like to call it as semi untargeted, semi targeted data analysis. And then reason we are going to see how and why. So again, lipidome or lipids are a part of metabolome. So and they are quite abundant. So you can see, like Tim just mentioned, like you can see lipids in our metabolomics platform as well. So they're not going to be as many lipids as we see in our lipidomics platform, but they are still there. Actually, there are a lot of them in metabolomics platform, and they're not very well identified. Most of them are unknowns, but we know there are, like, major classes of lipids that we do see in our metabolomics package as well. Okay, so the outline of this talk is going to be, I'm going to talk about the lipid class distribution that we've already covered, but I'm just going to briefly go over it again. And then the workflow for our data processing and data analysis, where we are going to talk in more detail about how we identify these things and then how we quantify lipids. And then I'm going to show an example data set and the report, basically a report that we give if you submit a data set, if you submit some samples to the core. So this is the class-wise distribution in the pooled plasma. By pooled plasma, we mean we have pooled plasma from Red Cross that we run for all our experiments here. It's like our quality control sample for our platform, like how our platform is performing from day to day and from experiment to experiment. So it gives a good assessment of, we know, like, what we see in this and how the quality of the data is. So it kind of serves as a quality control for each of our experiment and our platform in general. So in pooled plasma, positive, negative mode, most abundant classes in positive mode is TG, and in negative mode is PC. Some of the classes some of the compounds we do see that appear in both modes. And then, the way we pick it is the one that is more reproducible, looks more stable. We can pick one of the two from the classes when we want to do the combined analysis. So just wanted to point out a good resource for lipids. Lipid Maps is a good resource. This was like the first internationally accepted classification system for lipids, and it's a good resource for all the information related to resource. It gives the structure, there is structure drawing tools, it's a structure database as well. And then you can search just based on masses, and they have various tutorials, pathway information. You can you have like drawing tools for pathways and stuff. So it's a good resource for any information related to lipids. And this is just to point out the way we classify our lipids and the way we give what is the compound information, at what level do we give the lipid information. So in lipid maps, the way lipids are classified, each lipid has a category and then the main class and subclass. And this is the final identification for a lipid. So each lipid is a geometric isomer, and we don't have any information in between these lipids. But generally, on a more higher level, if you see, then we can group this lipid into further classes, in further levels. And so what we give in our lipid species identification is at this level, at the species level, where PC is phosphatidylcholine, is the name of the class, and then 36 is the carbon number of carbon, and then 2 is like 2 double bonds. So this is the information that we give in our reports, and the fatty acid scan species. So this information over here can have, there can be multiple combinations, and there is a way that, we can check these things manually, but right now in our data report, we don't give exactly what the scan species are. Okay, so the data analysis, and we have emphasized this enough in all the talks so far, but the data analysis actually begins at the study design. So this is the first and foremost important step for data analysis, is the study design. We actually start processing our data the moment we design the study. And the reason is, even before we get these samples, when we get these samples to run, before we run these samples, we have to take into account what are the factors, how the samples were collected, and if there is a time course in the data, and how big is the data set. We don't want to run let's say all the disease in one batch and all the healthy people in another batch. That itself will create a lot of variation. So you need that kind of information up front. And for example, if someone comes to the core with a set of samples, and then they want to follow up after few months with a different time point data, that's not going to be very useful if you come if you run a completely different time point of the data after a few months, and then you try to normalize these things, because you've already created that kind of effect, and it's very hard to get rid of that kind of variation in this. So first step is the study design. That itself, even before we run the sample, that's the first thing we have to look at, because that's how we decide how we are going to, what is going to be the run order of the samples. Then after we have this run order, we have to start thinking about various sources of variation. What are the various sources of variation we can have? It's a big data set. Like if we are running 600 sample over a course of 30 days, we are going to have a lot of variation just because of the number of days, instrument conditions. They're going to go up and down. Then if multiple people are prepping it, or different times we are prepping it, we are going to have that variation as well. And then what can we do to control these variations? How can we control? Whatever we can control, we should control this and take it to study design. But what we cannot control, we have to figure out a way to correct for these variations. And that's where normalization comes in. So why do we want to normalize? We want to normalize because we know that we are running these samples. We are going to have some variations. So we want to access these variations and correct them using a correct normalization method. Okay, so like I said, when we think about variation, there can be different variations. First variation can come from the type of sample that we are using. There's different tissues. You're taking different amounts of tissues when you are preparing your samples. So you have to see if they weigh different, and that's what we have seen when we use different weights to normalize. That works really well for tissues. Then urine, usually use creatinine to normalize. Then cell lines. Cell lines can have a variation too. People use protein content, or usually use cell count for normalization. Then analytical variation could be sample, because of sample prep, instrument capacity, and then drifts due to different batches. And then we use internal standard, our quality control samples. These are like replicated samples that we run throughout the experiment, like after every few injections. So once we take care of these variations, this improves our downstream analysis. Actually, after this variation, taking care of these variation, only we can get to the downstream analysis. For QA/QC, what we use, like I said, internal standards. This is a mixture of internal standard, but I think TM showed all the internal standard that we use. And we spike this mixture in all the samples, including our primary controls and the secondary controls. The primary controls I already mentioned, the pooled plasma, the Red Cross plasma that we run for each and every experiment. And this is like to get the overall data quality, instrument conditions, and variation from experiment to experiment. And the secondary control samples are our test pool samples, and this is the pool, like we take small amount from each of the sample for that particular experiment and create a pool of it. So it serves as a quality control for that experiment alone. So this we use to get the data quality and variations for the experiment under that under consideration. So if we get some samples from the client, we take small aliquot from all the samples and then create this pool and then inject this after every 10 runs or so. So for data processing workflow, once the client fills the sample submission form, then we go through the study design, and then we randomize these samples for run order, and then we run them through the mass spec, and then we get MSMS data. And here with this MSMS data, we run this MSMS through Lipid Blast software. So Lipid Blast is in silico library of MS fragments, and we try to match this against this Lipid Blast database. We try to match each and every sample with this Lipid Blast database and get a hit for all the lipids that we see in all our sample files. And then once we have this, we create like a master spreadsheet, like a master library for all the identification, whatever we see in each one of them. And so till this step, we use MSMS just to create the library, like a master library for our that particular experiment. And we take the mass, and then we take the retention time, and then the compound name, and then we create this library. Once we have this library, then we search this library. We search like a recursive search again through a targeted software, like a targeted software. We use multi-quant for that, and then we quantify this on MS1, and then we get the peak areas. So for MS based identification, so this is what the in silico Lipid Blast fragment library looks like. So the below one is the Lipid Blast from Lipid Blast. So this is for one of the internal standard, which is PC340. And like TM mentioned, this PC has a characteristic fragment of 184. So this is a major characteristic fragment peak of 184. And then, these are the different fragments for the same compound. So, and this is the experimental, coming from an experimental sample. So the way we match this is the scoring is based on how many fragments we matched from this and the major fragments, if they are like important fragments or not. So as you can see in the experimental ones, we have a lot of fragments, but the major ones that we have to match should match here, 184 being the most important one, because this defines the class, and the rest of them to call it that particular compound. So this is like another view, and I just wanted to show like what each of these fragments, this is how each of the fragment is defined like 184 corresponds to what, and then 492 corresponds to what, which part of the identification, and this is how it's matched. This is how we match in the database and get the identification. Yeah.
So, do you know that the library is using the same MS? Like, if you're using CID, do you make sure that the library uses the same thing? This is how you match the fragment ions, or I want to know why the fragmentation that you get is different from the library. Is it because they're using another type of fragmentation?
What do you mean by different fragmentation?
So you're missing some of those, right? Some of the fragments.
Yeah, because this is experimental. Ideally you want to see everything like right on top of each other. This is in silico based. So this is like, based on pattern. It's an artificially generated, like, based on the pattern. We have generated this is not like a real spectra from the compound, not from internal standard. But it's based on the pattern, how the classes elute and what the fragment is these are the fragment pattern. And ideally, yes, you would want to see everything, but experimental is not always ideal, right? You want to match maximum. So if you get the major ones, then this is the right hit, but not ideally, we would not see all of them. Okay. So yeah, this is another example for PE 340. This is also our internal standard. So we know this one shows like a neutral loss. So some of them, they don't have like a characteristic fragment, but they have a characteristic neutral loss of like 141 here in this case is a characteristic neutral loss for this one. Then once like I said, once we have this library, and when we do the second time search, we go for quantification based on MS1, and the peaks are integrated based on the MS1 scans. So now we have the peak areas and we have the raw data, and once we get this data, then we want to do our QA/QC analysis. Then we go to then we find out what are the variations and what we need to correct for, and then we go to normalization. After that, we do data pretreatment, scaling, transformation, depending on what we are doing here, and then we go for univariate, multivariate analysis. So, like first step for checking the quality control QA/QC is checking our internal standard. We spike these internal standard, and if we've run this over a few days, then we want to check for the days itself, like how the internal standard is behaving, and within the day also how the internal standards are. So this is the first step where we check the internal standard, because this is what we are going to use for normalization. So for normalization, the easiest and the most efficient way is to use the internal standards and the relative response to internal standards. That's what we use. But the batch effects are, when we see batch effects or some other, other drift effects that we are not able to correct with internal standard, there are other methods that we use. And for lipidomics data, mostly we use a method, Lewis, which we are going to talk a little bit more in the hands-on session, and there are regression models to adjust for batch effects. Okay, so this is just to show one of the example data sets, and what we were. So in this data is not. I don't think this has been discussed so far, but this was a data. These are like two different experiments over here, and then they were submitted at the same time. So I'm just using this as an example report, and the LRT are low low running capacity rats, and I guess HRT is the high running capacity rats, and they were trained for some time, and each of them are like, n is equals to 8. They were trained for some time, and few of them were, I guess 4 of them were control and then trained for 8 weeks, and then same in HRT. And then for running capacity, another set of running capacity rats, we have different age rats. So 4-week, 14-week, and 126-week old. So for the first training response study, the aim is to find the effect of training in this versus this rat, which is low capacity and high capacity. And then second aim for this particular running capacity rats was to identify if there is an effect of age on these rats, and then if this age effect is different in the low capacity runners and high capacity runners. So for this data set, so first step, like I said, first step is to just visualize the data and see how the data quality is overall, and visualize all data at once, like multivariate PCA is a good way to see how your quality control samples are. So these are the primary controls, the pooled plasma, and then these are the secondary controls, the test plasma, and then the rest are the samples. So this gives like a broader view of like, how the samples are, like what the quality of the data is. This looks pretty good, we can say, because the quality control samples are together. And then with this entire data set, we do see some kind of grouping here already. And then we check for the internal standards, RSDs, how the internal standard RSDs are. These are the internal standard RSDs. Everything is like below 25. So that's just one way I check and then use the internal standards only that passes the RSD criteria. And these are the internal standard we use for negative mode. Then we use the internal standards to normalize, and now then you view again like how it changes your data after you normalized we normalized using internal standard, and you check how the data looks now in terms of the quality. We see these things, they are more tighter, and then we do see the patterns are still there. Some probably a little more variation we can see more clearly, but the data quality looks good. And so then with the the QC plots, we also give this spreadsheet which has the first tab is the data information that is submitted by the client, and then we give we assign our sample IDs to each of the sample, and then the file name, and the researcher, match it to the researcher ID submitted by the client, and the run order, we give the run order in case people want to do their normalization or anything else. Then we give this run order for the samples. And then the next tab in this Excel file is the raw data, which contains the names and mass information, class information, the RSDs in the quality control samples, and what is the retention time, and then for each sample we give the peak areas. And then we also give the normalized data for, like, we already provide this data. And we also give like a combined positive and negative data, normalized data that is there. And the reason is, like I said, we see a lot of repeats, lot of species that we see in both positive and negative mode. And people often come to us and ask this question that, which one they're supposed to pick. So now we started like, combining this, and I give like a combined clean report where I try to filter out most of the false positives, or like, the noisy peaks that I see are not so good. So overall, people know, just directly can go to this and this, look at this combined clean data set. And for each of the experiments here, like I said, discussed before, we do ANOVA analysis, and the first analysis that we did was on two factors. One was the line is described as whether they are LCR or HCR, and then the training, whether they were trained or whether they were controlled. So it's so we did this ANOVA, and we give the p-values, we give the p-values for individual factors, and then the FDR corrected p-values for each of these lipids. So so we just look at the training capacity, rats, and you can see here the LRT controls separate from the rest. So the low running capacity rats, they are separated here from these are the blue ones. They are separated from the rest, and we see the same trend in our clustering here on a heat map, and which kinds of gives us an idea that the rats that were trained were kind of were like looks more similar to the high capacity runners as compared to the rats that were not trained. They are still very different from the rest of them. And then the second study was for the age study, and there we see that age has a big effect on these rats, and both on high capacity and low capacity runner. These are the young ones, and the 4 weeks old, and they separate, cluster together from the rest of them. And the same observation we see on our clustering. We see these are the 4 weeks, and these ones were 14 weeks, and then 126. So they separate pretty well over here too. And then this is just another way to visualize which ones are young ones. Like, this was just without defining which ones are LCR, HCR versus, and this is the same, but we just defined which ones are HCR and LCR. So not that much separating here, but the young are together, plus the old ones are here together. And this was like only for the lipids that were significantly different in for age in LCR versus HCR. So these are the lipids that differentiate these ones, and we see only one of two of these, like, cluster together pretty clearly. So this is just the general overview of, like, the basic report we give to the clients, because we don't know what exactly everybody is looking for, but we just give a overall information with some QC plots, with some unsupervised analysis, and I can take questions if, yeah.
I have two technical questions. So firstly, for the run order, you said it depends on the study design.
Yeah.
So is there any kind of guidelines on how to decide the run order?
Oh, so you don't have to, if you're sending it to us, you don't have to worry about that you have to decide that run order. But yes, there are things to keep in mind. Like I said, there are a lot of things to keep in mind. like how big is your study and what conditions are there, like is there a time course, then you need to take into account that you don't run all the time points, like one time point at once and other time points at two. So you don't want to create any variation by yourself. That's why, so when you're randomizing, you need to keep in mind that you don't put any sort of variation, like bias, by just putting them in a certain format. So that's why you need to randomize them.
Yeah, so is that like completely randomization or you have other tricks?
That's what, it's not complete randomization, right? That's, you have to block for certain things. You block first on certain levels, if based on the study design. Like I said, say for example, I have 200 samples, right? And then I have 5 or 6 different factors. So I want to make sure, like first I have to know what is the instrument capacity, right? Per run, how many can I run? If I have 200 samples and if I can run 50 at once, then I need to decide how I'm going to distribute all those factors. And I don't want to distribute, I don't want one factor altogether in one batch, right? So that's why you need to take care of those things before you decide. Not always completely randomize.
Second question is about the normalization methods.
Mm-hmm.
I just don't quite understand. You say relative response to internal standard,
Mm-hmm.
and then you listed different things. I just don't understand these.
So in general, so I said relative response to internal standard. In general, for metabolomics, we're just talking about lipidomics is a part of metabolomics. We're just talking generally, in general, when you do internal standard normalization, you have to, you don't have actually in metabolomics an internal standard for every compound, right, for untargeted. Same is the case for lipidomics, but it's better, but we have classes here. So we know where to go for particular, like for particular class. For PC, we have an internal standard PC, right? So we use PC for PC. That's the general idea. But in metabolomics, or generally, that's not the case. You don't have that. There are no classes in metabolomics. So what we do is you pick based on, you can pick based on your retention time, right? The compound which is nearest to the retention time of the internal standard. So use the internal standard that is nearest to all the compounds that appear in that region that you can use. Or if, so it's like you have to try and see what is the method that works out. But that's the general approach. You can use retention time based, or you can use RSD based, like the ones that reduce your variation that you can use. So that's why there are different things that I have listed, because you don't have class information always.
Okay, so will you, so in end of trying different methods, will you pick one, or you will apply multiple times of normalization based on?
For you mean just lipidomics?
Yeah.
Yeah, so generally, like if you have all the, like I said, you need to check before and after normalization how it's changing your data. It's important to check, right? You just don't apply it and say, oh, this is. So you need to check how it's changing your data. So you have to check your data before and after normalization. You have to see how your QCs are changing when you're doing the normalization, how overall your data is looking. Like PC is a good way to see how it's changing your data overall. So you apply different methods and you see which one is looking best, and that's what you pick.
Oh, so you pick one.
Yes, you pick one.
Not like I normalize it once and then apply another normalization method. You won't do that.
What do you mean? Like
you normalize it.
Normalize it multiple times, you mean?
Yeah,
Yeah.
according to different choice.
Normalize it different times if it's required. Like I said, if say if it's a tissue data, right? And I know that we get different weights of the tissues when the samples are submitted. So you have to decide if you want to, normalize it by weight or by protein. So in that case, yeah, you have to do two-step correction. So in that case, we do both, like, normalize by weight and also normalize by internal standard.
I think we're going to go into some of that when we do the hands-on exercises. And as a matter of fact, we're going to use one of the data sets that we're going to use is the one that Tanu just showed you. So we're not going to do all factors in the interest of time, but we're going to do some. Do you have another question, Karen?
I had a quick question. So it seemed in the heat maps and then the PCA showing separation on age that you were looking at age really more as a categorical variable, but I'm guessing it's just an artifact of visualization. So I work more with observational cohort studies and not always experiments. So any comment on that, or?
So I'm using it as a categorical variable. You're asking why?
Well, it looks like from the visual representations, the heat map and that you're taking continuous variables and making categories.
Yeah.
For example, with age, is that always true? And is there
No, it's not always true because they were on the same age. Like, the mice were exactly same age. That's how the data was given. That's why they were.
So maybe in the workshop we could talk more about situations where you have basically an observational cohort, not an experiment.
Right. So you're saying if you have like a range of ages.
Yeah, or a range of exposures.
Right. George, are you going to talk about that at all? All right. Yeah.
Thank you.
So, but Karen, I think it's a good point because it's quite common, right? So you often have to adjust for that, and then basically have to deal with that. Right.
Yeah.
We can definitely talk about that offline and maybe touch on that. And once again, I think we're going to use the same the data set that Tanu showed, although we're not going to look at quite as many factors, but we'll look at some. So anyway, I'll be happy to take further questions.